Life is never linear. Oftentimes it's circumstance and haphazard, an opportunity that just pops up randomly that one can never anticipate.
Tell us a little about your company's mission and kind of where it fits into the sort of growing field of precision health.
This is less about understanding how you and I are different. We're actually quite similar, but how our disease processes may be different. Biology is anything but reductionist and simplistic. It's the way in which we've been forced to do things from a simple matter of we can't understand complex data. It's hard to use it clinically. And from a regulatory perspective, this needs to be sort of controlled in a very precise manner. We know a disease is a very dynamic process as we go from normal to subclinical disease to clinically apparent disease to a therapeutic with regression of disease or a continuance of it. All of that is changing over time.
How do you think AI is going to change beyond where we are today?
How much is it truly improving the diagnosis of patients or the development of drugs? And that question has been a little bit uncertain. But what I will say is that this space is changing so incredibly fast.
Hey, Mo, welcome to the show today. How are you today in beautiful San Diego?
Life is great. I cannot complain and really excited to be here today, Chris. Thank you.
Yeah, I've been excited. You're fascinated by your company. I thought I would just want to get to know you a little bit and learn sort of what inspired you to create Sapient Bio.
Yeah, it's a fascinating question. And so this goes back quite a ways, and I'll give you the short version of it, but this has been a 20-year process. I was a clinician for many years. I practiced adult medicine and clinical cardiology, and one of the challenging questions that always would come from patients is, why did I develop this disease? And how do you know I'm going to respond to this particular therapeutic? And obviously, there's a textbook answer that we can always use. But one of the challenges of practicing in Boston in the Longwood area is that you get a lot of professors as patients. And they can smell when it's a superficial answer pretty quickly. And they'll challenge you with the next question and the next question. And it turns out for the vast majority of diseases, we really have a very poor understanding of sort of the disease pathobiology and even less of an understanding as to who within that specific disease is going to respond to a particular therapeutic. And so I became very excited and interested in the idea of being able to quote unquote personalize therapies. And really this comes down to how we understand disease processes. I had the pleasure of being at the Broad Institute in Boston for my postdoctoral work at the time of the genomic revolution. When we were finally able to parallelize sequencing, was able to see sort of the impact that sequencing had in the first wave of understanding disease biology, but was also able to very quickly see what the limitations of the technology were. It would have certain sort of influence in particular disease states, say oncology, but would be less useful in other diseases, and became very excited about other technologies then that would allow us to extend beyond genomics in understanding patient populations, being able to understand disease, stratify individuals, to be able to say, yes, we all have this same disease, whether it be diabetes or heart disease or cancer or Alzheimer's, but there's different subgroups within there. And being able to do so on a very discrete and quantitative level. And that ultimately is what gave rise to my academic career and subsequently the spin out of Sapient.
I remember those days, Mo. I think are you referring to those days where you remember the race to map the human genome? You had Human Genome Sciences and Celera, and we thought that if we knew the full complement of genes, that it would suddenly just cure all these diseases, right? But it didn't, did it?
It didn't. And that's the absolute truth. And I can remember very clearly a conversation with several heads who were involved in that race. And the conversation went like this, that if we were able to parallelize this technology to the nth degree, and we lined up every person on the planet and they held their medical records in their hand, and we could sequence every single person's genome instantly across the entirety of the planet, how much of human disease could we explain? And you can do that math. You can model that. And the answer is we want that to be 80, 90, 95, 99% of disease. The true answer for most common acquired diseases, whether it be heart disease, cancer, Alzheimer's, liver disease, GI illnesses, lung diseases, etc., for most common diseases, the answer is somewhere in the order of 15 to 20%. And so you're trying to solve a puzzle where you only have 20% of the pieces. And you can imagine how that is a very complicated sort of endeavor.
Well, that's probably a great lead into the mission and what you're currently doing. Tell us a little about your company's mission and kind of where it fits into this sort of growing field of precision health.
Absolutely. So the way we think about precision health, Chris, is that this is less about understanding how you and I are different. We're actually quite similar, but how our disease processes may be different. And the reason for that is the way we think about disease is that we start as quote unquote normal. And there's a process by which we go from a normal to a disease state. And then ultimately, what ends up happening is we're measuring that disease based upon that end pathology. And that's what gives rise to a diagnosis of heart disease, of atherosclerosis, or breast cancer, or Alzheimer's disease. It's based on that end stage, that end pathology, how a tissue looks under a microscope typically, or through imaging or other such studies. The problem is when we impose a therapeutic is not typically targeting that end stage, but is targeting the path by which you went from normal to abnormal. What ends up happening is if we think about these complex diseases, once again, whether it be obesity, diabetes, heart disease, cancer, Alzheimer's, all of which are quite complicated, we lump these all together and we say there is one disease and therefore there is one therapeutic we should use. We know, in fact, that's not the case. Empirically, we see this all day in the clinic. There's some individuals who respond wonderfully to a therapeutic, and there's some individuals, unfortunately, a good number, who have zero response. And that doesn't make sense. And this has to do with the fact that there's many paths by which you can go from normal to abnormal. And being able to understand that path, not from the end pathology stage, but be able to mapping that way in which people go from A to B, and then to be able to leverage that information to subset patients and to say, well, because you took path A, you're going to respond to drug X. Because you took path B, you're going to respond to drug Y. That's the essence of this. Now, genomics has done a tremendous amount in advancing our understanding not only of human evolution, but disease states. But remember that for the most part, the genome is stable from the moment of conception. It doesn't change over the course of our life. Now, in certain diseases, such as oncology, the genome is highly dynamic. And that's why sequencing has had great impacts upon those disease states. But if we look at everything outside of oncology, and even within oncology, if we look at diseases such as immunotherapy and those sort of transformative approaches, those are not leveraged so much by the underlying genetic mutational spectrum. They have to do with the expression of other aspects of the actual biology. And this is where thinking about how we go beyond genomics to understanding proteins, what we call proteomics, or small molecules, what we call metabolomics and lipidomics, has been absolutely essential. And so that's the genesis, if you will, of Sapient, using alternative technologies called mass spectrometry to be able to assay those additional information layers. And in order to do so, or using that information then, in order to understand both disease states as well as how we personalize therapeutics.
So Mo, before we jump a little bit more into your platform and your work at Sapient, so you were in Boston and then now you're in San Diego. Tell us a little bit about your personal story, how you went coast to coast here, and I guess as a co-founder of Sapient, you incorporated the company in California. I mean, tell us a little bit about that journey.
Absolutely. And I'll admit, Chris, life is never linear. Oftentimes, it's circumstance and haphazard, an opportunity that just pops up randomly that one can never anticipate. I grew up on the East Coast, right outside of New York City. Spent 20 years in Boston. My personal family is right outside of New Jersey, New York, Boston. My wife's family is right outside of Boston, and so for all intents and purposes, we were going to stay in Boston forever. I loved Boston academics. I had the pleasure of training at Brigham and Women's Hospital, Harvard Med School, MIT, and the Broad Institute. Absolutely loved every single aspect of the academic environment there. And that's where I was going to stay. And my wife personally is also an academic professor of immunology. And so that's what was going to happen. During the Obama administration, as part of the stimulus grants, my wife was awarded a very large stimulus grant to establish a genomic institute here on the West Coast. And so the plan had been that she would come out here for a year or two. This was before we had kids. We'd go back and forth. It'd be super exciting. We had never really spent meaningful time in California. I had no idea how to surf. The idea of surfing made no sense to me. And, we came out here, and as part of those visits, it became very clear that San Diego science was exploding. There was a very special feeling here, a very special culture that enabled technology development as well as cross-fertilization among very different disciplines in a way that Boston science may not enable in the same way. And because of that, we just became very excited. We ended up coming out here in 2013 as part of our faculty positions. Both my wife and I took independent faculty positions, started building our labs. And then in the midst of COVID, we said, why don't we, spin out a company? That sounds like a great idea. How hard could this be? Which is perhaps the most naive thought I've ever had. And and that's what gave rise to Sapient. And here we are today. We were lucky enough to take on a single strategic investment early in 2021, and from there have been growing ever since.
I'm glad I asked that question. That's a fascinating story. And I love sometimes how, if you're open in life and somewhat opportunistic, look at the doors that can open. So that's a great story. So tell us, Sapient Bio is often described as, and I know AI is sort of a core part of your platform, and but you're seen as kind of a leader in this AI driven biomarker discovery. Can you tell us what that means in simple terms?
Yeah, the way we think about this conceptually, Chris, is that, disease is complicated, and we're oftentimes forced to reduce it down to a single measurement, a single story. If your blood sugar is elevated, therefore you have a diagnosis of diabetes. If you have a plaque in your artery on imaging, therefore you have a diagnosis of atherosclerosis. If you have a specimen in pathology that has a certain appearance under a microscope, therefore you carry a diagnosis of pancreatic cancer. And we know biology is anything but reductionist and simplistic. It's the way in which we've been forced to do things, from a simple matter of we can't understand complex data. It's hard to use it clinically. And from a regulatory perspective, this needs to be sort of controlled in a very precise manner. One of the key aspects that's come out of the genomic revolution is just how complex human diseases can be and how dynamic they are. And so being able to measure not one or two markers, but thousands of markers at the same time, and then leveraged advanced information systems, what we call AI today. We used to call this reductionist statistics or Bayesian statistics. We used to then call it machine learning, and now we call it artificial intelligence. But the concept hasn't changed. The whole idea is that we're not so simple, and disease is not so simple that you can diagnose it or understand it based upon a single measurement, and rather we need complex measurements that capture the complexity of biology. And when we're able to make those measurements and then make sense of that data using these information technologies, it then unlocks the power to better understand biology and again, appropriately align a specific individual with their disease state with a particular therapeutic.
So is it correct to say that what you're doing is sort of looking at or using AI to look at a constellation of biomarkers and then trying to identify which are sort of predicting various types of disease, or whether it's prognosis or response to a drug? Is that, or are you also looking at, just identifying novel biomarkers that might predict some response, or is it, can you tell us a little bit about that? Is that the right way to think about it?
Yeah, that's exactly the right way to think about it. And whether or not they're known markers or novel markers actually just turns out to be a detail. In the end, it's exactly what you said, that we are able to now, instead of measuring, for instance, one protein in a tumor sample using immunohistochemistry, we measure 10,000 plus proteins in that same exact tissue section using mass spectrometry. And now we have the power to say, among these 10,000 proteins, which ones are critical for this disease state, which one denote sort of malignancy versus, benign, which proteins within this tumor now actually tell us who is going to respond to which particular therapeutic. And that's how you begin to leverage that type of data.
So Mo, we've heard, and I think most of our listeners might be used to like using biomarkers to kind of select patients for like a clinical trial. But I mean, biomarkers can also be used to detect disease earlier, right? So I mean, how do you see how biomarkers could change the way doctors diagnose or treat patients in the future?
Yeah, absolutely. There's no single utilization for a quote unquote a biomarker. It's simply a measurement that has biological value.
Mm.
And so when we think about this, it can be anything from early diagnosis of a disease. It can be prognostic markers telling us how someone is going to have a very sort of indolent disease state or have a very malignant disease state. It can be something as quickly as aligning a particular disease state to a particular therapeutic, saying this is why you developed this disease. And it may be different from the next person who developed that same disease, but for a different reason. And because you developed the disease for this particular reason, you therefore need this particular therapeutic. And so it's all of those. It goes from early diagnosis to understanding prognosis, to substratification in patients, to what we call companion diagnostics that allow us to align the disease again with a particular therapeutic. And then ultimately on the back end, to monitor for recurrence of that disease as well as particular response to that therapeutic. How do we know the drug is doing what it should be doing? And then how do we know over time if the disease has come back or if it's gotten better?
So could you maybe help us understand like the application. So, is there a real world example or case study, use case of how your research has helped uncover a new insight into human health or disease?
Absolutely. And there's hundreds to thousands of these. I'm going to give you just very simple examples by which we can do this. And you can see how this then can extend to virtually any disease. So we here at Sapient, we operate as a fee-for-service contract research organization. So we're in support of the pharmaceutical industry and the biotech industry that does the very difficult work of developing drugs. And our job is to help them understand their diseases and their processes so they can develop those drugs faster. So let me give you some very tangible sort of examples. So one of the largest questions that exist in the world of oncology therapeutics today is what is the actual best target to approach for a given cancer? Developing drugs has actually become much, much, much easier over the last 10 years, particularly with the newer generations of therapeutics, what we call ADCs or T-cell engagers or radioligand pharmaceuticals. Developing the chemistry of the drug is no longer the challenge. The real challenge now is what protein do I target in this specific cancer? Mm-hmm. What do I develop the drug against? And that's where the bottleneck currently exists. And so here at Sapient, what we can do is we can assay these thousands of proteins in a particular tumor type, and we can say this is the actual best protein to target because it's in the cancer, it's not in other normal tissues. This protein is expressed in the actual cancer cells, but not in the stroma. And this protein is localized to the cell surface, which means it can be engaged by a biological therapeutic, whether it be tied to an ADC or T-cell engager or other type of therapy. And so we can then identify what is the actual best target. We can develop that companion diagnostic so that can be used as part of clinical trials. And ultimately, we can use that same marker, if you will, to monitor for disease progression as well as drug responsiveness over time.
Yeah, I think it's fascinating, Mo, because I think many people think about the genome being like the most important thing, right? But I think that what many of our listeners may need to realize too is that it's the protein that's sort of translated from the genome that actually results in the function, right? That's why the proteomics is so important. If people think, many people may not have heard that term proteomics, right?
Yeah, that's exactly right, Chris. And again, we're forced oftentimes, just given the complexity of the system, to think in very reductionist and linear ways. And so if you go back to sort of the canonical thinking of biology that we learn in college, it's DNA gives rise to RNA, gives rise to protein, gives rise to function. And that's visualized in a textbook as a very straight line where the DNA, there's a single arrow to the RNA, there's a single arrow to the protein, yes, single arrow to function. And we know that's just not how the system is set up. There's levels of regulation at each of those steps. Fundamentally, what 95% of drugs plus are actually targeting is the protein. Now, what we've typically done over the last several decades is to use the DNA measurement or the RNA measurement to infer the protein levels, simply because that was the best we could do. Measuring proteins is remarkably complicated. What's happened in the real revolution over the last several years is that there's been a massive transformation in the underlying technologies that enable us to measure proteins or the subsequent level, which are the metabolites and lipids that emerge from those proteins. And that's really where we're based here at Sapient now. Because we can make those technical measurements, we no longer need to use DNA and RNA to infer, but you can actually measure the target of the therapeutic. And in doing so, you can imagine the fidelity of that measurement goes up tremendously because you're not using inference, but you're actually using direct measurement.
Let's just very briefly touch on that a little bit, because I know mass spectrometry is sort of one of the key tools that you use, right? And can you maybe describe in simple terms like how that works and how it's maybe different than from some other methods of detecting protein levels?
Sure. I'll start by just explaining how we measure proteins today. So typically we have technologies now that allow us to measure 1 or 2 or maybe a dozen proteins. They typically use an antibody to bind a protein, and then in some way there's a detection system on the back of that antibody. And it varies a little bit if it's a Western blot or if it's an ELISA or if it's an IHC, but fundamentally, conceptually, that's how it works. There's a binder that binds your protein of interest, and then it lights up in some way, and that's how we actually measure proteins. The challenge with those systems is twofold, or manyfold, actually. One, you have to know what you're measuring. It's not used for discovery, but you can do this only in a sort of targeted, known way. Two, you can only measure very small numbers of proteins because for each of those, you need a binding agent. And three, the fidelity of that measurement is really based upon the specificity of that binding agent, that antibody or protein to your protein of interest. We know there's challenges with all three. The sum space of what we don't know is far greater than what we know. And so to suggest that we know what the best target or best measurement is oftentimes that's not true. And antibodies are anything other than perfect in binding proteins. And so this has been extremely challenging in order to measure proteins as a whole, which is why technologies like sequencing enabled us to measure DNA and RNA to use those to infer protein levels. What mass spectrometry does is these are these big instruments in which we can put in very complex specimens, whether it be blood or urine or CSF or tears, eye fluids, tumors, tissues, cells in a dish, preclinical samples, virtually any sample type that's made up of thousands upon thousands of these different proteins. And in that biological specimen, now we can actually separate out those proteins and we quantitate each protein individually without having to know what it is up front. So there's no binding agent that's used there. In doing so, you can now go from saying, hey, I have to just measure this one or two proteins to saying, I want to measure all the proteins in the system and let the data tell me what are the most important one or two systems, or one or two proteins. And then we can develop an assay using mass spectrometry that only measures one or two, but does so in a very quantitative and accurate manner, as opposed to having to use antibodies as part of IHC or other types of measurements.
You often highlight your human biology database, and so what can you describe the kind of what's in that database and how is it different from a typical other, biobank or real world asset that maybe some other companies may have?
Yeah, absolutely. So there's a number of these databases in the world, whether they be sort of real world databases from large providers, networks, whether they be databases such as the UK Biobank. And each of these is typically based upon scale and cross-sectional analysis, where there's hundreds of thousands of people in these large databases, and you're assaying disease at a single time point. We know a disease is a very dynamic process as we go from normal to subclinical disease to clinically apparent disease to a therapeutic with regression of disease or a continuance of it. All of that is changing over time. And when you assay the system in a single cross section, you miss a lot of that actual granularity. And so we've built here at Sapient an internal database that we refer to as dynamic, given its dynamic nature, in which we've taken tens of thousands of people and we've serially collected samples from them over time. And what that allows us to do is to understand that progression and be able to molecularly characterize, well, these are the markers that tell us if people are going to develop disease X or Y, or this disease is going to progress in this certain way, or this individual, given their biology, is going to respond to a particular therapeutic. Now, we've done that for a number of diseases using human plasma samples from these individuals, as well as biopsies that have been taken serially in samples, whether it be across cancers, across normal tissues, et cetera, et cetera.
Interesting. So, you're one of your biggest customer groups is our pharmaceutical companies, right?
That's correct.
So they sit on massive amounts of biological and clinical data too. So like in your view, what's the biggest reason they struggle to turn that data into insights, and how do you kind of help them?
Yeah, it's a great question, Chris. And this is where there's a bridge between having samples, having the correct data, and then having insight. And each one of those, there's a gap between sort of those areas, if you will. Now, what pharmaceutical companies typically have is large numbers of samples along with clinical information. What they're missing typically is the molecular characterization of those biological specimens at scale. And that's where Sapient plays a role, where we can bring these samples in from a pharmaceutical provider. We can attain those protein measurements across thousands of proteins, across the phosphoproteome, the glycoproteome, understand which proteins are at the cell surface, understand what may be the best therapeutic targets, et cetera, but do broad characterization on a molecular level based upon the protein levels, the metabolite levels, or even the genomic levels. And then from that very complex information, draw the key insights, right? And ultimately what we want to be able to say is that this protein denotes individuals that have this certain disease biology and are going to respond to this particular therapeutic. And so going from just having samples to having molecular characterization to having true insight that allows you to substratify patients is how we typically work with pharma companies.
just to build on that, Mo. So if I was a new pharmaceutical client of yours, how would you typically engage with me? Are you bringing me a menu of options and assays, or like an or a full end-to-end biomarker program, or a specific access to data? I mean, how would you tell me a little bit how you engage pharma when you have a new client?
Yeah, absolutely. The simple answer, if you will, is all of the above. And no two clients are sort of created equal in that they all have their own very custom needs. Now, the way most CROs operate is that they're typically assaying very simple types of measurements. They're doing it at scale, so they build in some efficiencies, and there's margin that comes with that. Sapient is a different exercise in which we operate at the other end of the extreme, whereby we don't engage typically in very simple assays. Other people do that much better than we do. But rather, we operationalize very complex assays that are very custom. So, what I would say is no two pharma clients are the same. For some pharma clients, we're operating or working together with their early discover and target identification teams to help them identify what are the absolute best therapeutic targets to go after in oncology or in MASH or in Alzheimer's or in renal disease, irrespective of the disease state. In other pharma clients, we're working with them at a translational state where they have some preclinical information and they want to understand if what they've discovered in a mouse model or a dog model or a non-human primate is going to translate into the clinic. And so we can assay those preclinical samples. We can compare them to our internal database and we can say yes or no, this is the key marker in this preclinical system, and this is going to validate when you go into the clinic. And then for other clients, we're operationalizing and working with them on a clinical study level where they say, listen, we don't need to measure 10,000, but I need to measure these 4 proteins, and these need to be measured really well at a super sensitive level in a regulatory framework so I can understand and monitor in my clinical trial if my drug is doing what it's supposed to be doing. And so we can work with each of these groups. Our workflows are very customized to the client. We bring services so we can align with their samples and we can generate that data. We provide biocomputational and informatics support around these very complex data sets where we can begin to say, we will analyze the data that we generated for you and help you identify what are the key patient populations that responded to your therapeutic or did not respond. And on the back end, we can bring data to bear on these questions all the way from beginning to end. And so we like to be able to support our pharma clients all the way from soup to nuts, whether it be from early discovery through translation, through clinical, through data generation, through statistical analysis, and validation using internal data assets. And so this is about how do we accelerate the drug development spectrum as a whole. And in order to do so, we like to be able to fill in as many of those gaps as possible.
That's fantastic, Mo. I think for the remaining time, we're gonna dive deep here now and talk a little bit more about AI. Sure, there's a lot of potential ways you could utilize AI in the work that you do, but maybe let's start. Could you just describe where does AI and machine learning sort of sit in your tech stack today?
So if you look across biotech and life sciences as a whole, where the vast majority of AI is being applied to today is in a very small and narrow section around chemistry.
Mm.
And the question is, this is my target. What is the best way to develop my drug in order to hit that target? And that's where 80-90% of the AI applications are. The real problem that you're trying to solve there is rather than screening a million different peptides or a million different compounds to figure out which one hits my target, can I use AI then to optimize very upfront what are the best binders or develop new binders or new drugs that hit the target I already know? Makes a ton of sense, very effective, but it only solves the first or one of the first problems in a whole spectrum of issues around drug development. And that's precisely why you have not seen AI have the impact as a whole as it should. If you only solve 1 out of 10 bottlenecks, the whole system does not get faster, right? You alleviated one problem. Go ahead.
Yeah, no, I've just yeah, because I was going to be a little provocative and say that yeah, I mean I've just been following the headlines the last 2 years. You've seen pharma invest, I think, billions with AI companies trying to do exactly what you say, and I don't know, and I don't know everything, but I mean I've been following, and I haven't heard of anyone disclose like, hey, here, look at, we've discovered X number of new potential candidates or leads based on these AI discoveries or applications. Are you aware of any in your?
Yeah, there's more than a several of these that
Sure.
have and are matriculating through the system now, or either in clinical trials or will, undergo FDA approval. But fundamentally, the system hasn't gotten faster or cheaper or more effective. Again, you've solved 1 out of 10 problems. It's problem number 3, which is an important problem around chemistry. But unless you solve problem number 1 and 2 and 4 to 10, the whole system doesn't get faster or cheaper. And this is why the effect, if you will, of AI has not yet been felt. And in no way do I want to poo-poo this. This is, it's definitely effective and it's solving that one problem. And there are a couple therapeutics that are coming to market, but it has not yet had the impact that it should. And so where we're focusing a lot on the applications of AI are around that chemistry. How do we use AI to identify the absolute best target? How do we use AI then to validate that this target, in fact, is going to work for this exact modality? How do we use AI to understand the preclinical system that may be best aligned to the clinical disease state? And then ultimately, how do we use AI with foundational models, particularly in the proteomics or metabolomics realm, to understand the complexity of human biology and to be able to identify subpopulations of individuals who are going to respond best to a particular therapeutic? Now, when you align what's being done around chemistry with the upfront target identification and validation, preclinical testing, translation, and clinical trial implementation, this is where you now see the entirety of the system begin to move much faster and to work in a much more effective manner.
So Mo, if you look ahead a couple of years, I'm not going to put a time on it. I would say 5 years, but I mean, that's my God. I don't know what the world will look like in 5 years, but let's just say in a few years, how do you think AI, not just even the AI we have today,
Yeah.
but how do you think AI and multiomics will favor or how it will most fundamentally change how drugs are developed? Like, let's just focus like on the drug development process. How do you think it's going to change beyond where we are today?
Yeah, absolutely. It's a great question, and I'll admit that I have been a slow adopter in that I've always been quite dubious of a number of these systems. They look good that you can play around with them, you can make cat videos and do those kinds of things, but really when rubber hits road, how much is it truly improving the diagnosis of patients or the development of drugs? And that question has been a little bit uncertain. But what I will say is that this space is changing so incredibly fast. And in my lifetime, and I'm approaching 50 here, I've had the pleasure of working in the biotech or science space for 20, 30 years of that time. I don't think there's been another revolution here, if you will, either on a hardware or data side or software side that is accelerating and advancing at the pace that AI is. And so what we can do today, we couldn't do 6 months ago. And you certainly couldn't do it a year or a year and a half ago. So to understand where we're going to be in 5 years, I think is so incredibly complicated because it's hard to put a lens when something is so dynamic and changing. What I do think is going to happen is that we're going to move very quickly from single marker diagnosis. You have an elevation in glucose, therefore you have diabetes, to a much, much more holistic understanding of disease through multi-marker measurements. I'm very excited to see that the FDA and federal regulatory boards are starting to sort of jump on this very quickly. They understand this is imperative for improving human health, for patients, as well as for therapeutic development, for drug development, for our biopharma industry. And so that's one aspect that's going to change massively. Two, I suspect that because now of the application of data generation and AI, not only to chemistry, but again, for target identification, validation, translation, and clinical trial implementation, that the entirety of drug development is going to start seeing a real shift, and one that we haven't seen for the last 30 or 40 years. It costs more to develop a drug now than it did many decades ago, which is fascinating despite the advance in technology. It's, we call this Eroom's law, the inverse of Moore's law. And now I suspect with really systems now that can generate data at scale, in the AI systems that can leverage that data at scale, we're finally going to see a reversal of this. And drugs are going to be developed faster, cheaper, and more effectively, which you can imagine has enormous implications for human health and for the well-being of people, but also the biopharmaceutical industry as a whole. And I suspect 5 years is a frame in which this is very much going to come to reality.
That's good insight. I was just want to ask you. I think I've been, of course, biomarker discovery FDA's been asking for it. Pharma, it's the holy grail to help make, improve the success rates of clinical trials, to enrich the right patients in the trials. This has been going on for a while. So why is it that we're still talking today in 2026, Mo, about bringing down drug development costs, enriching patient populations, finding the right patients? Why is that still going on today? I just your comment just spurred the question, because I this has been something we've been trying to do for at least 10 years, if not longer.
If not longer. And it's not a new idea, certainly. The question is, how do you actually do it?
Mm-hmm.
Right? This has been the fundamental issue. AI is not magic. It's only as good as the underlying data that's giving rise to the underlying understanding. And we have not been able to, frankly, generate the right data that has the right impact in doing what we want. So whereas the concept is certainly old, and we've been saying we have to do better, right? The world needs to be better at developing drugs. We have to be able to deploy these more effectively. They have to be cheaper. They have to work better. They have to have less side effects. And the overall system has to move quicker. We have just not had the systems that allow us to generate that data or to be able to analyze that data at scale until very, very recently. And when I say very recently, I'm talking about the last 12 months. And so this is something that is certainly at an absolute inflection point. And it's really exciting to see now how these technologies, both on the data generation side, as well as the analysis side, are going to finally realize some of the promise of what we call personalized medicine. And so your point is very well taken. This is not a new idea. We've been talking about it for decades, if you will. You can go back to the Obama State of the Union, where he announced the Personalized Medicine Initiative, and nothing really has changed. At least we don't see drug costs coming down. We don't see drugs being necessarily much more effective. We're not seeing less side effects. And it is my true hope now that as the data has caught up to the concept, as the analysis systems have caught up to the underlying concepts, as the regulatory boards now are catching up to what these technologies can actually do, we'll see the needle substantially move here in the next year or two.
Well, that's a good message of hope, I think, and it makes a lot of sense, Mo. Maybe as we close out today's episode, I was wondering if you could share, as a leader now of the company, what are the emerging trends, or what are the things that you're watching closely that you believe might shape the future of the industry, and how is Sapient positioning itself to adapt in this era of AI? It's a very fast changing AI.
Yeah, it's a fascinating question. I think there are some advantages to being a small, quote unquote, startup. And certainly we've been around now for 6 years. So we still like to feel and think of ourselves as a startup, though we may not sort of qualify, if you will, as a startup any longer. But there's some real advantages to being small and being able to move in a very quick manner. And you can imagine when you have a 10,000, $100 billion pharmaceutical company, that's really, really tough to do. Change management is the hardest component of this in many ways. It's not the actual implementation of technology. And so this is where we're able to move much faster. The second part of this goes back to an earlier comment, in that the bottlenecks in this system have completely changed overnight. And this has to do with not only AI, not only with data generating systems, but how we fundamentally make drugs, in that it's no longer around a chemistry issue. The chemistry issue has been solved. And so if you speak to most pharmaceutical executives who work in this space, they say, just give me the target. I'll figure out how to drug it. That's not the problem. I just don't know what to drug. And so there's been a huge shift in what the actual fundamental bottleneck is. What that enables us now is, with our data generating tools, with our AI systems, is to apply them. You can point that spear to a very different problem. And if we do that effectively, again, this is where you see the needle moving in a very substantial manner. And so just to put it very tactically, the application of these proteomic or metabolomic or lipidomic or these AI systems now to target identification and validation, to understanding preclinical modeling, to ensuring effective translation and biomarker measurements as part of clinical trials, and then ensure effective clinical trial implementation, both in enrolling the right patients, so knowing who exactly to enroll in the trial, and then being able to monitor, is the drug doing what it's supposed to be doing in these patients, and how does that align with outcomes? That completely transforms how we develop drugs. And even today, 90% of therapeutics entering into the clinical trials are failing. You can imagine, if you cut that number in half, what that means for a whole, not only for humanity and well-being, but also for the economics of drug development. And so I'm truly excited for where we stand right now. I think needles are going to be moving in a very meaningful way here over the next 5-year period. And the implications for humanity are great because of that.
Well, we'll have to make that the last point of the show. I mean, fascinating. I mean, I'm leaving this episode for just energized by what's possible, and congratulations to you and all the great progress you've made in 6 years. It's a relatively short time, but yet so much work has been done, and I just wish you continued success, Mo. It was a pleasure having you on the show.
Thank you so much, Chris. I look forward to coming back in a year or two and talking about all the wins.
Yes, we should definitely check back down the road. So I'll definitely invite you back, Mo. Thank you.
Thanks so much, Chris. Have a great day.
You too. So that brings us to the end of today's episode. If you enjoyed this conversation, be sure to follow RX for Biotech on your favorite podcasting streaming platform, and subscribe on our YouTube channel to listen to more episodes just like this. And finally, if you or someone you know has just received a difficult diagnosis, especially a cancer diagnosis, we know that it can be overwhelming. And you hear the words, you hear the diagnosis, but you're left with lots of questions. Well, therapyforme.ai helps you do the journey, helps you take that next step. It helps empower you with the right information to know what questions to ask your providers to help you find a specialist near you that can help. There's no long waits, no guesswork, just clearer answers and a path forward. If you're looking for care, alternative treatment options, or just to find a specialist, or just need to be connected to a nurse navigator who can help answer some of your questions, start at therapyforme.ai. Thanks for listening.